Well, hello everybody, good afternoon. Welcome at the first Startup Forum on green, circular and sustainable innovations. We're going to bring an exciting program. We have 5 startups that are going to compete against the best startup of the EFIB 2020. And you audience are going to play a big role, obviously, in this, because you are going to vote and will get presentations of the startups. You just saw that passing by, went a little bit fast. We'll get back to the individual startups. But the idea is that everybody online will listen carefully. We're not going to make it too complicated. We actually reduce the complication to the minimum, and we're going to ask you to give 1, 2, or 3 points to each startup, in which 3 points is the best. And in the chat function, you will see a link where you can give your points. And also on the website of the EFIB program, you will find that same link. And then we have our session this afternoon on circular and sustainable innovation. We get 2 more later this week. And on Friday, around 2:30 in the afternoon, we'll be presenting to you the winner. What will the winner get? First of all, a lot of attention from the media, social media, all kind of publicity, also from Europa Bio and EFIB channels. So that's a good boost for their knowledge. And also for DSM, because I'm speaking on behalf of DSM and Planet Bio. I'll explain to you that later. But they also will get a 1-hour coaching session from an expert, either at DSM or at Planet Bio. So, so far the process on the startups, I'll take just a few minutes to introduce who is, oh yeah, thank you. There I get the slides from the startups. No, I get the slides from the jury. Also good. As you see, we have Michael Krel from Sofinnova Partners. We have Rob van der Meij, maybe or maybe not, he wasn't there yet. Zhou from Bits and Bytes, and we'll ask a few questions, but please audience, join us in thinking about nice questions. Put them in the chat function and we'll try to ask those as well. Yeah, so I was back at Planet Bio and DSM. Just a few slides for the introduction there. I'm the managing director. I worked for 14 years at DSM in several functions in R&D and innovation, always around industrial biotech. And 2 years ago, I got this really exciting opportunity to develop the industrial side of DSM, which has been in Delft for 150 years already, to turn this site into an open innovation ecosystem. Next slide, please. Yes. So the ecosystem is called Planet Bio, and we would really like to be the innovation hub for industrial biotech. We're located at the Biotech campus in Delft, and we're doing basically 2 things, offering in a nice building, good labs, offices, and a nice environment for white biotech companies. So you can physically locate with us, but more importantly, we create a total ecosystem around white biotech, industrial biotech, synthetic bio, in order to make sure that the companies that are with us in Delft don't only with us, but they really manage to grow, and they really manage to get to that level of creating impact and really moving our economy to a bio-based economy. And we have plenty of things to offer. So the next slide, you can see that. Oh. Here. Just a summary, but well, we are in the west of the Netherlands. That's a nice thing because there's a lot of universities around, a lot of schools, a lot of talents, and a lot of good fun, also nice cities. Traditionally on the site we have enormous amount of technical skills, competences in analytics, in food application, in fermentation, in scaling up. We are very close to the Delft Technical University, and we have a strong collaboration with them on courses and with people exchange. Then we have DSM here also on our location, and that's our sort of big father or mother. Actually, they're independent. We are independent as ecosystem, as campus from DSM. But if people want to have a door into DSM, obviously it's very simple to create that. We also have the bioprocess pilot facility. That's a pilot plant. Some of you may know that plant here, also on campus. Very simple for the people that are working here to just take a few steps and look at the options there. For the rest, it's just a nice place. We have a restaurant, a gym, everything you need, and a lot of space. And particularly also, and that is what we believe makes us quite unique, we have space for scale up. So we still have greenfield, we still have a place for industrial activities. At the same time, we're in the middle of Delft, so that is super unique. And just nice for the people, for the companies that are with us, to support them in their growth, also in the phase of going to production. That is everything. No, then I'll do that myself. Next slide is our community. You see DSM, Centrind, BPF, the pilot facility. In the middle, Veramaris, Olatin, and Avancia are joint ventures. And at the bottom, there are our startups, and we are growing pretty fast. We will be very happy to meet more companies, because we're looking for everybody that has a business in industrial biotech to join the ecosystem. So that was my story. I'll keep it here. Feel free to contact me also on the partnering calendar if you wish. Today is about the startups themselves and not about our ecosystem. So I will move now to the jury members, so they can also quickly introduce themselves, and then we go for the whole program with the startups. Michael, are you there? Michael is from...
Yes, I am.
Nova.
Just now, I should be unmuted. So hello, my name is Krel. I'm part of the Sofinnova Partners team. Sofinnova is a large venture firm fully dedicated to life sciences and early stage life sciences. To give you a few numbers, we managed to take 2 billion euros, which makes us one of the largest player in that field. And so for us, life sciences are 3 different topics: pharmaceutical biotech, medical devices, that are the bulk of what we do. And for the last 10 to 12 years, we started industrial biotech activities, which has grown significantly, up to the point that we are now investing 125 million euro fund that is fully dedicated to alternative to fossil carbon for the chemical industry, and maybe more specifically, leveraging biology for sustainable production in 4 verticals: agriculture, food, chemicals, and materials. And we are based in France, investing globally. As for me, and I'll keep it short, I'm 43, I'm a PhD in organic chemistry. I spent a few years as a business development guy in industrial biotech startups and joined Sofinnova 7 years ago now. That's it for me.
Okay, thank you, Michael. That's all clear. Then I would like to move to Joseph and get an introduction on Bits and Bytes.
Thank you, Cindy. Hi, everyone. This is Joseph from Bits and Bytes. So Bits and Bytes is based in Shanghai, and we are also a big fan of industrial biotech. Just our main focus is more on the industrial biotech with the application on the agriculture and food industry. So our main investment thesis, including sustainable agriculture, animal nutrition and crop nutrition, and also covering the alternative protein, which is very hot in Asia right now. And we invest in early stage startups from pre-A, Series A, and Series B. And we have just increased our check size from 1 to 3 million. Despite we're based in Shanghai, we do have a global mandate that allow us to invest in the best companies around the world. But we do have a very strong Chinese market applications and hoping to bring everyone that we invested to the Chinese markets. That's it. Thank you.
Thank you, Joseph. And I'm happy to see Rob arriving exactly in time as well. Welcome, Rob. Rob, can you quickly introduce yourself and Capricorn to anybody that does still need an introduction on Capricorn?
That's okay. I'm Rob van der Meij. I'm a partner in Capricorn Partners. We manage several venture capital and listed equity funds. I'm active in the Capricorn Sustainable Chemistry Fund, which is investing in industrial biotech, but also classical chemistry to try to create better products and better technologies for the world in terms of resource efficiency, sustainability of the product, or the use of the product. We invest both early stage and later stage. We invest across the world. We have several investments in the US, in Europe, as I said, both in early and late stage companies. I'm a failed engineer that turned into a venture capitalist. So that's still a good professional career track for people who are not detailed enough to be real engineers. I can recommend it to you. Thank you.
Thank you, Rob. So that leaves us then to the start of the pitches of our startups. We want to start with Juan Medina from Kaffe Bueno, located in Denmark. I see you started in 2017, already have 5 employees working, already have some interesting products that we saw, and a nice collaboration with Evonetix. So please introduce us to your company.
Perfect. Thank you, Cindy. Hi. Well, good afternoon, everyone. My name is Juan Medina. I'm the co-founder and CEO of Kaffe Bueno. We are an ingredients company based in Copenhagen. So what we do is that we upcycle spent coffee grounds for ingredients for nutraceuticals, cosmetics, or the food industries. Over 9 billion kilograms of coffee are consumed every year. And only 99% of this, we only use 1% of this, and the other 99% ends up in the waste. This is a huge waste of resources, as coffee is so good as a beverage that we have totally forgotten that it's actually a bean that has polyphenols, lipids, sugars, proteins, and lignin. All these are high value compounds which are key for the up-and-coming bioeconomy. Our focus is on tapping into the known health benefits that the coffee brew has and related to the specific compounds that are within the coffee. So we take a more holistic approach into the coffee and see it more as a bean rather than only as a beverage. So how it works is very simple. We upcycle it from industrial waste producers. It can be hotels, office, coffee chains, or big industrial producers from instant coffee. Then the problem with coffee is that after consume, it's pretty wet and moist, so it needs to be stabilized quickly. The first patent that we have filed is for a biopreservation solution that we put in the spent coffee grounds, allowing us to extend its shelf life, to call it that way, so that we don't need to pick it up every day or every 2 days. We can pick it up every week, every 2 weeks, which obviously allows us to have a very flexible supply chain, but most importantly, keep that food grade material of the spent coffee. Then we process it in our biorefinery, where we use different green chemistry and biotechnology from green chemistry, enzymes, water, pressure, different temperatures, and here we fractionate it into the different compounds. Then we sell them directly as it is, or we use it to produce other high value ingredients. We use it through distributors or directly to manufacturers. Our first product in the market is our Kaffe Bueno oil. We introduced it in the market until last year. Right now, we signed an agreement, a JDA, and also a commercial agreement with Evonetix for the application of this oil, of this product within the cosmetic sector. We have proven to have anti-aging, sun protection, moisturizing, and also anti-inflammatory properties. Also, as a food and beverage application, we have proven not only to have a very nice coffee flavor, but also to extend the shelf life of the product itself. Then we have our Kaffe, which is a small particle-sized microbead, which we ground even more finely than what actually coffee is. And these we're selling it into the cosmetic industry as well as a replacement to plastic microbeads. Then we have our Kaffe, which is being used by different bakeries and confectionery companies here in Scandinavia, which we have focused first. It's very high in antioxidants, has more potassium than a banana, so very good for keto diets. Very high in proteins, also a high water holding capacity, shelf life extension, and you don't need to use caramel color to add coloring into your end product. So it is clean label colorant for the bakery and confectionery industry as well. The team is composed right now of 6 people. Basically, the 3 founders, we originally are from Colombia. This is kind of a reason in our story behind this. Where we believe that farmers in Colombia were not getting paid what they should, but that's because companies here in Europe and in the US pay for only actually that 1% that they use. So obviously there's a big resource inefficiency that is exactly what we are trying to tap into. We have a combination of PhDs in biotechnology, health and nutrition, and also people working in our PR and communications. So it's a very balanced and growing team. We have a strong advisory board from people from within the cosmetic industry, such as Carl Lindner. Then we have Marcel Leuven, who is past DSM and Avantium. Then from DSM, Walter and also Steen, which is president of AAK and has a past as well in Christian Hansen. So they are all making sure that we are scaling up and tuning our ingredients into the right markets. Right now we have 4 patents filed. We need to file some PCT applications this end of the year, and the other one we have won several prizes. And currently we're preparing the launch for 3 new ingredients in Q2, Q3 next year. Some of them will be for cosmetics, and we have another one aiming for the food industry as well. So this is currently where our different ingredients are being used, mostly in Europe. We're looking to expand seriously into the US. We have been seeing a lot of interest from there. Our commercial partners, which are Givaudan and Norman, they are helping us in distributing our products to the right partners. Then we have partners from the engineering side, such as Alfa Laval. We are a certified B Corporation, and we have investors from Pauli, Becksfonden, Yield Lab, and different institutions here in Denmark. So we believe that coffee is not only one of the most consumed and delicious beverages on earth, but we believe that has one of the biggest potential in this up-and-coming bioeconomy, especially in Europe and in the US, where it's highly consumed. And what many people forget is the hidden emissions that takes to bring coffee into Europe or the US. So we hope to mitigate this emission by giving a better use to this biomass full with new applications and resources. Thank you very much.
Thank you, Juan. That was quick and impressive.
On point.
Members of the jury, you're all muted still. I don't see any hands up. Do you have questions for Juan? Yes, Michael.
Yeah, I can start maybe. Hi, Juan.
Hi, Michael.
We met already, it seems like a long time ago. I was wondering, I had two questions. One is about the application work and development work, because you have a few products that are extract from in a way raw extract from your coffee, and then you have to substantiate in a way all the claims, and especially the nutraceutical claims, and the good for the health and good for the skin and good for whatever. So is it something that you perform yourself, or is it something that you outsource, or is it something that you leave to some of your partners?
Well, it's a combination of both. Let's say for the cosmetic side, obviously for the cosmetic, and for the nutraceutical side, in vitro, so we could run it. We have run it ourselves, but also we have partners who run those in vitro studies and in vitro screening first. And obviously, as we go, let's say if it's a nutraceutical to be tested more in a rat or in an animal in vivo, that obviously this is not something that we do. We have partners here in Denmark and in Europe who help us on that aspects. And then on the cosmetic side, on the cosmetic is the same. We have partners such as Givaudan, which have an amazing platform where we could test these ingredients. So yeah, it's a combination of both, and it depends the ingredient itself.
Okay, Cindy, can I ask a follow-up question on that?
Yes, go ahead.
Okay, so following that question, and also coming to the partnership that you have with Givaudan, my understanding is that when you are in the ingredient space, your ability to support the claims and especially having strong R&D proof of the claims that you are claiming is a very strong value creation point. And so I was wondering, how do you see the value sharing, both from a distribution perspective, because I guess that having Givaudan as a distributor is a very powerful tool, but it as well is very costly. And on the other hand, having in vitro study, but not yet, especially on the nutraceutical, going up to the point of having human, not clinical trials, but close to. Do you see that as a potential evolution of your business plan going forward, or do you think that you will need to narrow down and focus on developing new extract and product from your feedstock? Thank you.
I think it's a combination of both. At the end of the day, here, maybe sometimes we're too ambitious, but at the end of the day, I think the key for our successful business in Kaffe Bueno is having a combination of both, some high volume, low value ingredients, to say it that way. Meanwhile, we have some smaller value that we can sell by grams, backed by patents and clinical studies. But I think it's a combination of both. It's really choosing, let's say, from the beginning, from all that pool of candidates, compounds that you have, to choose your winners. And at the end of the day, it's always not a bet, but an investment. So obviously there's the risk that it might not work as you want it to work in humans at the end of the day. But yeah, I think we have a nutraceutical compound that is moving to preclinical studies next year, for example, which we are very excited about. So yeah, I think it's a combination of both again.
Okay. So looking at the time, maybe if we have a very short one from Rob or Joseph, I will allow that.
Okay. Well, I just have a quick short one. We see a lot of coffee or spent coffee extraction companies coming up. Where does Kaffe Bueno distinct itself from? How do you differentiate, especially technology-wise?
Well, I think our technology in itself, it's unique. I don't know of any other company who focus entirely on the health aspect of the coffee itself. I think we have it pretty much more scale up than any other company as well. And obviously, it's not only about that technology, but also on that commercialization aspect to find a market for products and ingredients that are not yet in the market. It takes time, and to produce them also takes infrastructure. So I think either way, if there are, let's say, obviously there are going to come other companies, I think what we'll be doing here in Kaffe Bueno is a company that is going to be being stand up in the next 50 years. It's not the case, but I think our technology and the fact that since we are, let's say, further ahead, we have access to certain compounds within the coffee, which these companies have not yet been able to access in decent quantities to experiment good enough and create or discover new applications of them. So I think it's not only, of course, you have to produce whatever ingredient you produce at the most cost-efficient way, but again, it's in finding new applications to what you have, where more value is created.
Okay, that seems to be a satisfactory answer. So thank you. We'll leave it here. Everybody, still a small reminder of the voting. So you see it in the chat where you can vote 1, 2, or 3 and give us a total view on the whole company. Then we move to Stan. Stan is the director and chief scientist of BlueGene Technologies from the UK. Stan is more or less on his own, but he has several co-workers that work part-time for him. And he raised his company in 2015. Stan, the floor is yours.
Oh, hello there. Greetings to you all from London. Well, we actually have a nice day for a change. I hope the light and the sound is okay for you. Now, I'm pleased to introduce to you today my biotech startup, which is BlueGene Technologies. And I also would like to relate to you the story of bioindigo and how we believe that BlueGene Technologies can help contribute to our planet's health. This is basically the focus of the session and of the whole meeting. Good. I would like to take you back firstly to 1988. During my PhD studies at Imperial College London, I became the first person in the UK, if not in the world, to make the well-known dye indigo by genetically engineering bacteria. This with some genes that I had cloned. And you can see this picture on the right of the slide showing the original photo of the first clones producing pure indigo in large amounts. I realized at the time that this could be the basis of a future commercial biotechnology process. But at the time, everybody joked about my crazy blue bacteria. And except for some academic visitors, we call them academic visitors, because I don't believe they were, they promptly returned to Korea and started laboratories devoted to the production of bioindigo, as it later became known. So maybe it wasn't just a crazy discovery or bit of cloning. I wasn't just ahead of the game. In fact, I would say I started this game. So, moving on. Later on, in the 1990s, came some big players, and following in their wake, a large army of academic research groups who tried to produce bioindigo. But despite the big bucks, still in 2015, there was still no commercial bioindigo. Well, of the many possible reasons that we can speculate about why this hasn't happened yet, there are many other factors that come into play, such as the cost compared to synthetic indigo produced chemically on the industrial scale, and also people weren't ready for GM genes. And finally, no one had at the time heard about global warming, least of all the textile industry. Let's not forget. But since then, a lot has changed, and especially awareness about the environmental impacts of the chemical and textile industries. Thanks to increased consumer awareness, the textile industry is slowly waking up to reality and trying to adopt more sustainable and less polluting processes and products. But all along, I had a ready-made technology and a vision of the future that others had been trying to copy for the last 30 years. So there you go. 4 years ago, I formed my startup, BlueGene Technologies, and Imperial College's synthetic biology incubator in London provided the proof of concept funding and free lab space to begin the commercialization of my original innovation. As a result of rapid developments, Innovate UK identified BlueGene as a company to watch in 2017, and since then, BlueGene has gained 2 further European funding grants, one of which is currently being going on at the study is going on at the moment, funded by the Clean Enabling Technologies or Clean Production Consortium. Today, our versatile bio-conversion process is closer to fulfilling our original vision, which is now everybody's vision at this meeting today. That is to replace the outdated industrial chemistry of the last 100 years with a cleaner, safer, and sustainable alternative. And that is bioindigo. In other words, instead of all that toxic chemistry, why not let bugs do the dirty work? And what's not to like about it? It ticks all the right boxes. It's desirable, being 100% organic, no solvents or heavy metals are used, no toxic wastes or aniline residues. It's sustainable and it consumes low-cost bulk feeds. The chemistry is identical to the natural and synthetic indigo, so it plugs directly into the existing markets with no need for equipment and little change to processes. But the story doesn't end there, you see. The textile industry is still faced with the fact that all synthetic dyes being used today cause some form of pollution, either in their synthesis or in their application. How are we going to address that? One dye isn't going to change everything. And if you think about it, we've been making beer, wine, and vinegar using microbial fermentations for thousands of years. So why not brew our dyes as well? Wouldn't it be nice if we could make all our dyes by fermentation and not just indigo? Well, perhaps I'm beginning to sound a bit crazy, you think. Let me show you what I'm talking about. So 4 years ago, I started with just one dye, bioindigo, and an idea I wanted to test. As you can see, in this history of BlueGene, since 2016, using our versatile platforms, we've engineered microbes to produce a whole range of stunning and novel biodyes, over 12 in fact, and the number is still growing. As with bioindigo, they are supreme examples of green 21st century chemistry and synthetic biology. Unlike many natural biological pigments and dyes, BlueGene's dyes are stable, light, fast, and water resistant, making them ideal as textile dyes, but also opening up new opportunities. So it's not only possible to brew many dyes, it's possible to develop new ones as well. Is there any demand for our novel dyes? Well, to date, there is. We have identified a considerable number of potential clients with a genuine interest to test our products. Here you see some of them. Ital Denim is looking to be Europe's greenest denim maker and is requires 1,500 kilograms per week. English Fine Cottons is a high-quality textile manufacturer in the UK. Danish textile designer Kvadrat are probably the world's largest private a textile designer, industrial textile designer. Chinese textile manufacturer Oh-Jin has requested samples for testing. French cosmetics company L'Oreal requests samples for testing in hair and cosmetics. We also have a number of UK textile manufacturers and dyers, and 4 academic research groups looking for samples to test our products. Good. I beg your pardon, I'll go back there. I would just like to say our market research shows that textile manufacturers, retailers and dyers love our range of vivid organic colors, and for good reasons too. Since about 2010, retail chains like Zara and Walmart have been striving to improve their environmentalist image through sourcing more organic textiles. But the dyes they use are just the same as before. And then when it comes to natural dyes or organic
just a small question, Stan. Please take a note at your time, because your time is running out. And I see you still have 2 slides, so please speed up a little bit.
Okay. I would just like to say that our organic dyes are perfect complements to organic textiles. In short, there is a lot of demand, but we can't supply these potential customers yet. So this is where you, the investors, come in. In order to fulfill these requests for the growing list of potential customers, we are looking for investment to enable the scale-up and supply of these dyes at the kilogram scale. This is a necessary step for commissioning tests and validation by industry stakeholders to justify further investment and support for industrial scale-up. Importantly, investment will leverage further UK and EU grant funding. Finally, I would like to introduce the team behind our success. 4 years ago, BlueGene was just myself, some bacteria, and some DNA sequences, and an idea. Today, BlueGene has a portfolio of dyes and scientific technologies, and is supported by an exceptionally strong scientific team and advisory board, and also by textile industry and academic advisors. The team are ready to go. So I would just like to say, if you are thinking of investing in a biotech startup with a rosy or colorful future, please contact me through the EFIB after this presentation. And to complete the story of BioIndigo, I would just like to remind you on this picture that indigo is not only blue. Thank you very much for your attention, and I'd like to happy to ask any questions.
Thank you very much, Stan. I see a hand of Rob. Rob, go ahead.
Hello, Rob.
Hey, Stan, good to see you again.
Yes, nice to see
virtual.
you.
So, if you think about as investors in this space, we are obviously looking at potential investment returns, but we also look at impact. Do you have a bit of feel for quantification of what your solution brings on say, environmental footprint compared to the current commercial technologies?
Well, the current commercial manufacture of indigo, the industrial manufacture of indigo, as everybody knows, requires petrochemicals. It requires toxic petrochemicals, nitrobenzene, nitrobenzaldehydes, obviously solvents, a lot of heat and other components, heavy metals, catalysts, and finally the output also includes a large amount of toxic wastes, which is well documented in the literature. These include formaldehyde and some, yes.
But did you make an attempt to quantify this? Huh? Because
no,
as investors, we like to report to our shareholders.
Absolutely.
If we invest in this, there are so many kilograms of that being replaced by so much of this, and this is so much better for the world. That would, because I think you have a great story. I really think you have a great story.
Sure.
No, but if you can put that quantification to it, I think it will support your story much stronger.
I fully agree with you. Well, of course, that is a job for somebody else to carry out, and that can be done by some of the expertise lies in some of the partners of these European cooperative consortia. I think there's one in Germany, but I forget which one it is. So it's a question of time to do that. The bottom line is that we eliminate all of the heat, large amount of the heat and solvents. So and large amount of the chemistry that is involved. There's no chemistry required for the extraction as well. So the solvents for the extraction. So you're automatically reducing the whole carbon footprint of the synthesis.
Stan, I have to stop you here. I think we all agree with the potential enormous benefit of your bio-indigo, and I think we can open the invitation to anybody that wants to help you to quantify some numbers. Due to time, I need to move on to the next speaker, which is Stephanie. Stephanie is from Evonetix. I'm sorry. You're still in the startup session, Stephanie, but your company is already 58 people. So you're growing very, very fast. Founded in 2015. Tell us about the big secret of your company.
Sure. Hello. So thank you, everybody. So yeah, I'm just going to switch gears a little bit and talk about more an enabling technology as opposed to a particular application. So what we're doing is actually developing a gene synthesis platform, and this is a desktop platform which you would have in your own lab. So it's accessible to every researcher who's doing engineering. And why is this important? Because we all know, as we're hearing in this conference, there are many huge challenges that biology can solve. But currently, the way people synthesize their building blocks, which underpins all these applications, is too slow. So obviously, looking at all of the different applications, because it's an enabling technology, it's applicable across all of these different industries to help you rapidly prototype and get successful engineering experiments. So as I mentioned, currently how DNA is made and supplied is too slow. And this is something that we get a lot of feedback on, and we've spent a lot of time talking to a lot of end users across all of the different industries and segments. And currently most people outsource because this has been very much a cost sensitive aspect of engineering. And so they use a service model. However, the turnaround times is just far too slow. And there are other aspects as well, which limit people's ambition at the moment. So by building a desktop synthesis platform that synthesizes DNA at high accuracy, scale and speed, we will look to enable people to run much more ambitious projects. So we've developed not just a desktop platform, but this enables you random access to your own platform in your own lab. So you can use it for rapid prototyping whenever you want to. And really, we're hoping that this would become as widespread as NGS. That's certainly my background for the last 25 years in NGS, and we've seen technologies move back in-house by enabling them to become more accessible. So we're hoping to see the same again with gene synthesis. So this is a picture of our platform, and I won't go into too much detail about this in here because I'd like to tell you more about how it actually works. So we are using a chip-based method, so a highly parallel method to synthesize, many thousands of DNA molecules at the same time. We're using a thermal control chip, which means when you combine that with a thermally labile chemistry, it means that you have a very fast platform that can synthesize, many, many thousands at high accuracy across a chip. And because of the unique features of the technology, this thermally labile chemistry approach, we are also not just synthesizing DNA, but we're also automatically assembling DNA into long double-stranded DNAs on the same instrument. Because we use a synthesis approach which uses heat, we can actually do error removal during that assembly process, which I'll show you a bit more about. So this is our chip, and this is just to show our prototype chips. And so across our chip, we don't actually have physical wells. What we have is these virtual wells where we have columns of heat in a flowing liquid, where we can very discreetly control the temperature of that liquid above each of these sites. And that means you can achieve the high parallelism that's required by flowing the same chemistry over the whole chip, as opposed to delivering to individual wells. And as you can see from the picture on the end, that if you don't have the chip designed in the right way, if you apply heat to a flowing liquid, you create a kettle. However, because we have a very unique closed loop chip, we are able to discreetly create these columns of heat on each virtual site across the chip. And what that means is that on each site, we can independently, precisely control the heat. And by using a thermally labile chemistry, it means we can very specifically create thousands of different oligos on the flow cell surface. And then using an orthogonal chemistry, we can selectively release those oligos from each of those individual sites. And in the flow, we can enable 50-mer oligos to flow to the next site and allow hybridization to occur. And this is the start of the assembly process. And then what we can do, because we can apply heat, we have a machine learning algorithm that predicts the perfect match temperature, melt temperature for that duplex. So we then apply a temperature just below that, and any oligos that contain errors are melted off the flow cell surface and removed in the heat, in the flow. And we do this over and over. So we then hybridize pairs with pairs and pairs with pairs with pairs. So as we go across the flow cell, we generate much longer double-stranded DNAs. And at each part of that process, we're testing for the errors and removing those errors as we go. So we end up with a number of fragments across the flow cell, which may be, for instance, 2.5 kb long, or anything upwards towards 250 kb with a 10,000 site chip. And what that means is that we can overcome some of the issues that conventional approaches have, which is as you accumulate errors, you have a diminishing yield of perfect DNA. And because we're removing errors as we go, we actually can overcome that, so we can produce much, much longer DNA. Our philosophy is a low-cost platform with single-use cartridges and a cloud-based approach. So as and when you want to use different throughputs of synthesis, you use a different cartridge. We're also working on an enzymatic method as well as a phosphoramidite method, which means whichever technology is working the best at that time, we'll just drop that into this agnostic platform. So really, the market has told us that they want something that's much faster. They want to have rapid prototyping, and they want greater length and complexity of their DNA. So to achieve this, you obviously need something that's highly parallel and gives high fidelity DNA. So what we're going to do is box this up and deliver it into the hands of individual users at a very cost-effective and accessible price. So in summary, we plan to solve the 3 key problems at the moment, which really is a bottleneck for a lot of people in synthetic biology and engineering, is overcoming the turnaround times that some people are experiencing. So then they can synthesize at scale and high accuracy. And obviously they have full control over the technology because they have the platform in-house. And that's it.
Whoa.
Hopefully I kept the time.
Oh, thank you. That was blew us all away. Thanks for that impressive talk. Just a very, very small question. I think many of us have this question in the audience. Despite the whole technological part of your what exactly is the gain in time we're talking about?
Sorry, what is can you repeat that, please?
The gain in time. You say our competitors are much slower. How slow are they? How fast are you?
Yeah, so it depends. It depends on, you speak to different end users. It can take, a week to many weeks to many months. And we're talking about a day to a couple of days.
Okay.
Stephanie?
Yes.
Maybe a comment and a question. So first of all, thanks for this brilliant presentation. You showed a very interesting market, which we believe fully in, especially, and maybe this is also a disclosure, we are investor in one of your competitor or player in the same field named DNA Script that has somehow a similar approach, somehow only. I was wondering, so this DNA printer in a way that you are ready to release, when do you see that coming online and being able for Cindy to buy one and test it?
Yeah, so I'm fully aware of DNA Script, and basically, we're going to be starting our early access program towards the end of 2021.
And just a question about that, and maybe this is a dumb question, but given that you are still based on phosphoamide chemistry, is there any safety rules that needs to be applied, or because this is so miniaturized, then there's no real issue and you can put that anywhere? Just out of curiosity.
Yeah, it could be anywhere. I mean, everything's self-contained. But we are also working on an enzymatic method as well. So when we come to market with this, if the enzymatic method has progressed enough and it offers the greater advantages, then we will use that in our cartridges. At the moment, we're working on both because obviously phosphoramidite chemistry has been around for 30 odd years and we know a lot more about that. But yeah, we're maintaining a rather agnostic approach, and whichever one will win out will be the one that we go with.
Okay, thank you.
Thank you very much, Stephanie. Joseph, we haven't heard much of you. If you have a question, this is the time, and otherwise I will move to the next speaker.
It sounds very impressive. So thank you, Stephanie. But can you just share a little bit about your business model? How much are you going to be charged for this machine, and who are your targeted customer?
Yeah, so we think actually the biggest USP for this platform is being able to assemble long double-stranded DNA. So we think those who are doing, pathway engineering, metabolic engineering, folks who really are going to benefit from longer gene constructs are going to, that's going to be a sweet spot for our technology, because at the moment, that takes some time and effort to do for most folks. And as far as the platform's concerned, we're looking to go in at, around the 30K mark. So, it has to be competitive. We know that this is a very price sensitive space. So that's why we're not putting that barrier in the way.
Thank you.
Thank you. Thank you, Stephanie. We're moving to Borja, who is going to talk about his company, Xtrem Biotech, raised in 2013, coming from Spain with 2 employees.
Hello. I hope you can see me,
Yes.
hear me properly. Thank you for the introduction, Cindy. Yeah, 2 people company so far. We are a spin-off company, so have a good team of researchers behind from the University of Granada. We are a company working with extremophile strains isolated from hypersaline conditions, which gives them great capacities as a biostimulant and biofungicide for these 2 great markets, which are growing at 2 digits growth. Okay, as we have seen before, there is a quite difficult regulation abroad internationally, not only in Europe, but especially here where we have a real tough regulation. So, as I said, we work with these strains which were isolated from hypersaline conditions and have both capacities, which is quite not exclusive, but has allowed us to play or to sell and introduce the product firstly in those two markets, firstly in the biostimulant scene. We have at the same time a proprietary library of extremophiles microorganisms to reiterate and do this process again. But our first product is based in this XT1 bacteria, which as unique selling point, I will mention the high capability to of course, withstand salinity conditions is one of the problems that agriculture is facing more and more due to the use of mineral fertilizers or the use of underground waters. Okay, so these are some of the strain's characteristics, not only the strain, but what produces the strain, the metabolites produced by it are very interesting and could be used not only in agriculture, but other side industries that we are looking development with other partners. So our first product, the one we started to work with a local company here in Spain, formulator of chemicals, is this microbial liquid product, which has a clear biostimulant effect on root development and crop production, and enhances plant response to salinity, water scarcity, or pathogen stress. Okay, this is a product that we have been producing since the very start. We have achieved to even produce and upscale the process up to a 500 liters reactor inside. Not at a state-of-the-art reactor, I would say, thanks to the capability of this strain. Again, you don't need great requirements for the upscale. If we move into this picture, we can see how the right plants treated with the strain, of course, had a great development compared to the ones treated with water during 2-month trial. And this was scientifically, of course, tested and seen on the several parameters like height, weight of the area plants, or number of flowers for these two crops. Okay. Again, regarding the biostimulant effect, we should mention that it enhances the precocity of the crop. The crop will come into production sooner, so this will be an advantage in terms of price and access to the market firstly, where the consumer is able to and is looking to pay a little bit more than after 2 or 3 months that the crop is on the market. Okay. Secondly, we have to mention very interesting this seed treatment capacity that has been tested in greenhouse internally, and in a few trials outside. We will be soon able to test it with a cooperative from Germany, and where we can see here in the below picture how the plants, when applied a different dosage of strains per milliliter, had a significant difference in terms of growth, and again compared to a commercial product existing in the market. As I said, we are based on the scientific park of Granada, back from the university, and we are able to perform these in vitro trials from the start. It's not a product test only, so we are able to know how it performs, how it works, the strains, especially this XT1, where we have been putting our effort and expertise. Upper picture, we can see on Mac bean after 5 days how it enhances the germination of the strain, or below how the exopolysaccharide produced by the strain allows it to stick to the surface of the strain. These are other characteristics that can be applied to other industries. And thirdly, we are happy to have been part of a SME's phase 1 project from the European Commission, that allowed us to test and study the market of verticillium pathogens, especially in crop and olive production, where it's a great problem, a soil problem attacking olive trees and causing great losses on plant on trees. Okay. Here we can see how injected the strain is a strain that can be formulated or can be applied or restored at a great time for logistics or for co-formulation. Lastly, as a business-to-business company, we see or started our venture as a production and commercialization of biostimulant. We are selling and reaching our maximum capacity so far in terms of biostimulant and selling local companies. But we are looking for product development opportunities for new uses or presentation that are out of our hand in terms of upscaling or downstream processes. And of course, we are looking for ventures or capital to go forward on the licensing and registration or commercialization of the strain as a biofungicide into the plant protection regulatory. To finish with, I'm one of the co-founders of this company with microbiologists from in-house and outside the company, and strategically supported and backed by DNA advisory services. Thank you very much, and I'm looking forward to answer your question.
Thank you very much, Borja. Well in time.
Thank you.
That leaves space for 2 questions from the jury. I'm looking if any of the 3 has a question.
I can start. Thanks, and very interesting. One question I got was, I used to work for a company that developed Xtremophyte. And so a difficult thing about Xtremophyte is the way you scale up, because nobody knows how those will perform. Nobody knows how to engineer those, and nobody has the experience of scaling up those fermentation. How do you see that? Do you feel this is a threat for your development, or do you think that you have acquired enough knowledge about that to go further?
Well, I don't know what kind of Xtremophytes maybe you work with, Michael. I don't know if they were
Deinococcai bacteria.
Ours are mainly Bacillus, isolated from hypersaline conditions. For us, it has not been a great challenge to I would say. Of course, we had fermentation issues, everything is spoiled, and the foam produced by the metabolites were a challenge to address as well. But I don't see it was something difficult for us. Okay, we are not, we don't see ourselves as a company to invest in equipment in a 180 cubic meters reactor, as I said before, as I saw before in Biosphere. But I think we have a good knowledge on how to scale the strain. We have, as I said, we have achieved to do it at a large scale with another state of the art. I would say it was a reactor adapted from a BR production, a BR reactor. Okay, we achieved to do it. I myself had to enter into the reactor, if I have to say, to clean it once and to put biofilms aside, but that was the worst thing to do.
Thank you. A short one.
Okay, short one is, how do you see the commercialization timeline? Because the regulatory path is quite, could be quite long.
Yes. So far, as a business to business, we are a company. We are producing and selling to third parties, which are following their own regulatory or commercial strategy. But for the biofungicide, where all the value is, where you can recover all the investment on the patent, it's easy to have 2 years wait.
Okay, thank you. We have officially 11 more minutes left. So to stick in time, I'd like to move to the last speaker, which is Oh-Jin from Lignum from South Korea. Company was founded in 2017 and has 5 employees at the moment.
Okay, thank you, Cindy, for facilitating this presentation. I'm very honored to present our company and Lignum's products to European and global audience. As Cindy said, our company was founded 2017, at the end of 2017, so it's less than 3 years old. And I am in charge of marketing and innovation. And next slide, please. Lightweight composites are most sought after material in composite industry. And as the magazine and recent issues showed, the lightweighting is very important issue in especially transportation industry. And our company is tackling this lightweight issue in vehicles first. Next slide, please. Recently, media reports that Porsche brand made their motorsports cars from natural fibers. In 2019, they made 2 doors and rear wing, but this year they report complete body. But if we extend to low-priced cars, we have to think about differently. Next slide, please. We can ask, can bio-based provide alternatives or better ones in terms of price and also easy processability? Right now, polypropylene occupies the most part of polymer parts in automobiles. As you know, the production process generates more carbon dioxide than lignin-based materials, Lignum and also other companies are developing. So compared to conventional fossil-based production process, wood-based, lignocellulose-based process generate less than 1/4 of carbon dioxide. So it can give many benefits, not just lightweight, but also it's renewable and also carbon neutral. Next slide, please. So next slide, please. Okay, Lignum's tackle this problem with simple physical and chemical treatment to meet the same high safety and quality standards, and also in more economical and energy efficient way. So our process consists of 2 simple physical and chemical processes to generate bio-based fillers that can be used in automobiles and also other industries. Next slide, please. Our product have superior properties. They are simple process and renewable raw materials. And also our products can miscible with other conventional polypropylene, polyethylene, polystyrene, also ABS and polycarbonate. biofillers have good dispersion in the matrix. Lignum's biofillers have low density, and also they can be mixed with other biodegradable polymers, polylactic acid, and polyhydroxyalkanoate, and PBAT to modulate biodegradability. And Lignum's biofillers have a good processability. We already have our products on the market. We can supply biofillers and also biocomposites and master batches for the customers can use in their areas of applications. As for master batches, we can manufacture 60% of bio-based bio content. So they are available for other applications. Right now, next slide, please. We already used our Lignum's biofillers in passenger vehicles in Korea. There are 3 models which already use biofillers in tailgate and door trim. And we are also collaborating with global automakers for other parts, interior parts. As you know, even though we changed to battery-powered vehicles, we still be concerned about the lightweight. So Lignum's biofillers can provide benefits in gasoline-powered vehicles and also battery-powered vehicles as well. Next slide, please. Right now, we can provide 100 kilogram biofiller per day, but we'd like to build a demo plant by 2022. And if we are successful with the demo plant, we'd like to go commercial in 2026. We really welcome forestry industry to participate in this endeavor to commercialize this adventure. And we also welcome consumer products industry and also electronics industry as well. Next slide, please. Our team, 5 people, but here on 2 slides you have CEO and me, and we are moving to the next Series A funding. Next slide, please. Well, we are recognized by government and chemical companies and also social value companies. So with this product, we would like to move forward in the global arena. So I'm very glad and thank you for giving Lignum a chance to present. Thank you.
Thank you, Oh-Jin. Jury members, Rob.
Yes, Oh-Jin, thank you very much for this. Very interesting. If you look at the applications you have today, is that mostly in polycarbonate or ABS replacement?
Right now, we are focusing to polymers right now, polycarbonate and ABS, and also polypropylene. 3.
And what level of percentage replacement do you get in these products?
Right now, 5 to 10% replacement can give 5 to 10% weight reduction.
Yeah. So you mean you get 5 to 10% weight reduction?
Yeah,
or do you replace 5 to 10% of the product?
5% replacement with our biofillers can give 5 to 10% weight reduction.
Okay. Thank you. And when you look at cost for these people, how does that relate?
As far as the cost is concerned, we estimate It depends on the amount of order, but we estimate $2 to $3 per kilogram.
Okay. Okay, and can you tell us a little bit more about your chemical treatment process? Is that involving what type of steps are involved in there?
It depends on the raw materials. So as you know, there are different lignin products on the market, from paper pulp biorefinery and other straw-based biorefinery. So it depends on the different raw materials. But you are focusing right now on wood-based.
But do you apply the same? Okay, so do you apply the same process, but you just tweak a little bit in terms of conditions for each type of lignin that you get?
Yeah.
Yeah.
Yeah. So we are now in dialogue with several raw material providers in terms of supply of raw materials.
And do you think you can take any type of lignin, or do you say, well, I need this kind of range of specifications that I need to get here?
We haven't tested yet, but our process is quite flexible in terms of raw materials.
All right. Thank you very much.
Thank you.
All right. Thank you, Oh-Jin. And that brings us to the closure of this session. I enjoyed it very much. 5 wonderful speeches. Thank you for the presenters. Thank you for the jury also, Michael, Rob, and Joseph. Don't forget to vote, and can still be done over the coming days. And let's see who will be the fortunate winner on Friday. Thank you. Have an enjoyable week, and look forward to seeing you in another session.
All right. Thank you.
Bye-bye.
Bye-bye.
So Cindy, you can also access your meeting room, your online meeting room, if someone clicks in. So you just can go via this link in the chat and can meet.
Oh, yeah. So you're sending that now?
Yes. So it's in the chat. It was the first one. So we put a lot of them right now for the speakers and jury members. You just have to click. You just have to click on your own link. Do you see it?
Yeah, I see Borja, Stephanie, Stan, Oh-Jin, Juan. I don't see mine yet.
It's more above. So you were the first one.
Ah, I see. I'm sorry, I didn't see that, so I could not tell the people that they were able to.
I forgot it in the beginning, but
yeah,
no worries. So maybe there is someone you can just click in, and if there's someone you can say hello.
Yeah.
Okay.
Okay. Thank you very much. Bye bye.